Hello and welcome back to Anatomy and Physiology. This is video 2 in a 3-part lecture series over skeletal muscle. What we're going to be covering today is excitation, contraction, coupling. So today's lecture is going to be much more physiology intensive than the previous lecture. So the video 1 in this lecture series went over skeletal muscle anatomy with a focus on microscopic anatomy and the ultrastructural characteristics of muscle fibers, right? What's happening at the subcellular level? What are the organelles that actually allow skeletal muscle contraction to occur? Today we're going to be treading into the physiology, and that's kind of the area or the topic that I find most interesting. So, as always, I've provided you with a set of learning objectives. Follow through with them. Have your lecture handout either printed out so you can take notes as we go, or up on a computer so you can type your notes out as we go, and let's go ahead and do it. So there are going to be two main points we talk about today. We're going to talk about the nervous system control of muscles, and we're going to talk about excitation, contraction, coupling, what actually happens at a muscle. We're going to paint it around a story where somebody is taking a drink of water, and we're going to talk about the pathway leading from the brain, ultimately ending out at the skeletal muscle that's involved in contracting your muscles in order to grab, let's say, a bottle of water and take a sip, right? It's actually quite a complex process, but we're going to try and simplify some of the more convoluted portions of it for you today. Now, before we do that, we're going to do a quick review so I know that you understand what scale we're actually looking at. So, if you remember on this image, we have our tendon. Our tendon splays out and forms a network of connective tissue that surrounds an entire skeletal muscle. That network of connective tissue is called the epimysium or epimysium. And here we have our entire skeletal muscle. You can see those are big. We're looking at a cross section of the brachioradialis. Now, skeletal muscles themselves are broken into discrete bundles of fibers called fascicles. So fascicles are made up from anywhere from a few to many hundreds to potentially thousands of muscle fibers, depending on the size of the fascicle. The fascicle is surrounded by the paramysium, or paramecium or mycium, depending on who your instructor is. And then as we telescope out here, we've shifted from things we can see with the unaided eye, even though fascicles are really small, they're like the grains in meat, to the things that we can't see with the unaided eye. And at the end here, we see a skeletal muscle fiber, aka a skeletal muscle cell, and that's what we're going to be looking at today. We're going to be looking at a skeletal muscle cell in a tremendous amount of detail, and we're going to look at how the nervous system ultimately regulates the activity of an individual skeletal muscle fiber or skeletal muscle cell. Remember that skeletal muscle fibers or skeletal muscle cells are surrounded by the endomysium, which is like a network of areolar connective tissue that provides a little bit of electrical insulation. Within the muscle fiber itself, within the muscle cell, we have organelles called myofibrils, and within those myofibrils, they're made up of myofilaments. So down at this scale, we're talking about things smaller than an individual cell. And if this image is confusing you, go back to the previous video and take another look. Try and answer those questions from the previous handout. So another thing we need to review before moving forward today is just the structure of a skeletal muscle fiber or skeletal muscle cell. Now, I made an exaggerated point in the previous video of saying, hey, what we are looking at here is not a skeletal muscle. It is not a fascicle. We are looking at one individual muscle fiber, aka muscle cell, and we're looking at this cell very, very magnified. We're looking at it very close up, and we're looking at the smallest components in that cell, notably the organelles that allow for skeletal muscle contraction to occur. So the plasma membrane of a skeletal muscle fiber is called the sarcolemma. If you remember from the previous lecture, the sarcolemma has voltage-gated ion channels in it. The functional organelle, or the contractile organelle within the skeletal muscle fiber, of which there are many in an individual cell, is called a myofibril. Surrounding the myofibrils are T-tubules, which transmit electrical current from the outside to the inside of the cell, which is necessary for the sarcoplasmic reticulum, and specifically the terminal cisternae, to release calcium. Now, if we were to pull out one of these myofibrils and look from Z disc to Z disc, you have these structures called sarcomeres. Sarcomeres are the most important functional unit within a skeletal muscle because that's what actually produces contractile force and allows a skeletal muscle fiber to shorten and to pull on bone and ultimately move your body. When you think about an entire skeletal muscle, most of it is just individual little sarcomeres, these tiny little subcellular units that are so important for understanding the contraction of a muscle. Now, from Z disc to Z disc is a sarcomere. Coming off the Z discs are your thin filament. Coming off the M line are your thick filaments. Thin filaments are made from actin. Actin is the structural component of the thin filament. The regulatory components of the thin filament are troponin, which is indicated by 12, and tropomyosin, which is indicated by 11. So again, we've gone over that a few times at this point. And then you have the thick filament, which is made from myosin. And myosin proteins kind of wrap around one another and they come to this tip at a myosin head, which becomes really important in understanding how muscle contraction occurs. Now, there are a lot of complexities to the regulation of movement, but essentially what our motor systems do, our nervous system, our motor systems in our brain, what they do in our brain and our spinal cord, what they do is they mediate movement, and they want to do, or the goal is to achieve two things. You want to activate movement when it's the desired outcome, and you want to inhibit unwanted movements. The activation of movements when you want them and the inhibition of movements when you don't want them follow different pathways. There's what's called the direct pathway and the indirect pathway. For the sake of simplicity, when I'm describing the nervous system regulation of movement, I am going to focus on the direct pathway today. So here we have an image of the brain. And we've looked at this image. So we're looking at the cerebrum and we're looking at the functional mapping of the cerebral cortex. A couple of regions that become important for this discussion today are the premotor cortex and the primary motor cortex. Now, the premotor cortex is a really complex brain structure, and a lot of its functionality, it's broken into 5 different regions, and it's really hard to describe its function in simple terms. But for the sake of this class, we'll say the premotor cortex is involved in the selection of appropriate motor plans for voluntary movements. In other words, the premotor cortex collects information, processes that information, and uses that information from other brain regions to plan complex movements. The primary motor cortex, on the other hand, initiates movements. It's involved in the execution of those movements. And that's the pathway that we're going to spend a lot of time talking about today. Now, some other brain structures you need to have a grasp of. You need to just know they exist, and I'm going to condense and simplify what we're talking about, are these areas of subcortical gray matter. So they're deep to the cerebral cortex, and collectively these areas, these little bundles of gray matter, are called the basal nuclei or the basal ganglia. Now, there is a rather complex circuit that involves the substantia nigra, the globus pallidus externa, the globus pallidus interna, the putamen. We're not going to talk about all that. I am going to bundle everything in the basal nuclei, and I'm going to say it plays a really important role in smoothing out skeletal muscle contraction and preventing unwanted contraction. And we're not going to memorize this circuit, but suffice it to say, there's a little bit of complexity, right? There's some overarching regulatory mechanisms that allow us to contract muscles when we want to smooth out those muscle contractions, and to prevent unwanted contractions. What we're going to focus on today is the direct pathway of muscle contraction. So now we're going to paint a story, and we're going to start with our nervous system regulation, then we're going to get to excitation, contraction, and we're going to tie it all together. So let's say you had these particular items on a table in front of you, and I said, hey, go ahead and take a drink of one of those. So you have human urine, sulfuric acid, and a bottle of mineral water from Aquafina. Now, you may go with the human urine. I don't judge, right? I don't know your life. You don't know my life. I'm not going to judge. But most people are probably going to opt for the Aquafina. So that's where we're going to start. That's what I was talking about when I said I'm going to paint a story around taking a drink of water. So when you think about it, that information is going to enter along the visual pathways and it's going to be processed in the visual cortex. The auditory pathways, because I've asked you to take a drink, it's going to be processed in the auditory cortex. You're going to get multimodal association areas working in there. But eventually that information is going to make its way to the premotor cortex. Now, the premotor cortex communicates with the primary motor cortex, which communicates with the basal nuclei, which communicates with the thalamus, and then the substantia nigra just inputs to the basal nuclei and kind of modifies what it's doing. So let's go ahead and think about our muscle contraction. Step one, you've been given a set of instructions, and now that's bouncing around different regions of your brain that process that information. That information goes to the premotor cortex, and the premotor cortex plans that skeletal muscle contraction. It goes, the most appropriate muscle contraction set to carry out for this particular set of instructions would be to contract my muscles and to reach for the water. So the premotor cortex is going to communicate that information to the primary motor cortex, and that's going to activate what's called the direct pathway. Now, it may seem a bit indirect, but this is the direct pathway that regulates movement. And I am simplifying for anybody who's taken like the science of neuromotor control. I am simplifying what happens in the basal nuclei. It is an introductory class at ACC. I just want you to know that the basal nuclei are really important. So the primary motor cortex is going to initiate a skeletal muscle contraction, but first it's going to send that information to the basal nuclei. Right? So the information from the premotor cortex goes to the primary motor cortex, and the primary motor cortex goes, okay, I'm going to run that information about the basal nuclei. Now, the basal nuclei also has inputs from the substantia nigra that can inhibit unwanted contractions, promote and smooth out contractions. So there's inputs from both places, and in fact, these neurons in the substantia nigra play a really important role in the development of movement disorders like Parkinson's, but again, beyond the scope of this class. The basal nuclei is then going to communicate with what's called the motor thalamus, and the motor thalamus is going to have its inhibition relaxed, and it's going to communicate with the primary motor cortex. And you don't have to know that particular pathway in a huge amount of detail, but I do want you to know the basal nuclei and the thalamus play a really important role in regulating contraction. Once the motor thalamus, the inhibition has been lifted and it activates the primary motor cortex, now is when I want you to really start paying attention and thinking about a pathway you would draw. So you get the activation. This would be the cell body. So you have a synapse between neurons in the thalamus and neurons in the primary motor cortex. These neuron cell bodies you find in the primary motor cortex, these are what are called upper motor neurons, or UMNs. Now, the dendrites in the cell body of these upper motor neurons, which are also called pyramidal cells, and any tract leading from the cerebral cortex to the spinal cord producing muscle contraction is called a pyramidal tract. But these upper motor neurons, the cell bodies and dendrites make up the primary motor cortex. And that's what initiates skeletal muscle contraction. So diseases like ALS destroy the primary motor cortex. If you have no primary motor cortex, you can't initiate skeletal muscle contraction, and that produces paralysis. Really problematic, right? So this is a really important functional area of the brain. Upper motor neurons then have axons that project out of the primary motor cortex. These axons form what's called the corticospinal tract. The corticospinal tract and the axons of these upper motor neurons then cross over at the medulla oblongata at what are called the medullary pyramids. They descend down and they communicate with the somatic motor neurons in the anterior gray horn. The somatic motor neurons in the anterior gray horn are activated, so this is a glutamatergic synapse. The cell body of the somatic motor neurons is located in the anterior gray horn, but the axon projects out, becomes part of the ventral root, fuses to form the spinal nerve, travels along the ventral ramus in this case, all the way out to a skeletal muscle fiber. And the point at which the axon, right, comes to its end, the terminal bulb or the synaptic end bulb, as I'm going to call it, that's where you get communication between the nervous system and the muscular system. The point at which a somatic motor neuron communicates with a skeletal muscle fiber is called the neuromuscular junction. And then there's a set of electrochemical events that take place here that ultimately excite a muscle fiber and allow it to contract. So when you want to contract a muscle, premotor cortex plans, primary motor cortex initiates, upper motor neurons, which form part of the corticospinal tract, communicate with somatic motor neurons, right, which form nerves, project all the way out to the muscle fibers that they control, and they synapse with the muscle fibers at a region called the neuromuscular junction, or NMJ. That's before we even get into excitation, contraction, coupling. That's the nervous system regulation. So when we say skeletal muscle is under voluntary control, it means the message to contract that is coming from higher order brain centers that regulate conscious thought. Now, when you look at this image of the spinal cord here and you see these somatic motor neurons, these somatic motor neurons project out of the anterior gray horn and they branch. And they branch to control multiple skeletal muscle fibers. A motor unit is a somatic motor neuron plus all of the skeletal muscle fibers it innervates. That's what constitutes a motor unit. So when a motor unit activates, an action potential travels along the somatic motor neuron, and then it will activate every muscle fiber associated with that motor unit. So if you're looking at your practice problem, think about those identifying the motor unit, and then in order to figure out what motor unit is bigger or smaller, count the number of muscle fibers innervated by one somatic motor neuron. The muscle fibers plus the somatic motor neuron equal the motor unit. Now, if I ask you which do you think would generate more contractile force, well, motor unit 2 is associated with 2 muscle fibers. Motor unit 3 is associated with 3 muscle fibers. Motor units activate. So when this somatic motor neuron fires an action potential, it's going to activate all of those skeletal muscle fibers. Motor unit 1 would generate more contractile force because it just has greater muscle mass behind it. So, for example, if you want to generate more contractile, let's say that you're picking up something light, right? You may activate this motor unit 2, because you don't need all your skeletal muscle fibers. Then let's say you're picking up something heavy like your girlfriend's couch that ripped your thumbnail completely out, right? You're going to activate more motor units because you want a greater mass of muscle activated. And that's one of the important topics or points that comes up in the EMG lab. So when we think about simplifying this, premotor cortex plans skeletal muscle contraction, primary motor cortex initiates skeletal muscle contraction. Action potential travels along upper motor neuron's axon, which forms part of the corticospinal tract, right? And then you get a point of synapse between the upper motor neuron and the somatic motor neuron. Motor neurons are multipolar neurons, meaning that they have multiple feed-in points, but we'll just pay attention to one here. That somatic motor neuron, right, receives that signal at the synapse, transmits an action potential out a spinal nerve, which eventually becomes a more specific nerve, like the brachial nerve. And what's happening here is you're seeing the excitation contraction coupling that takes place at the skeletal muscle fiber. So ultimately, the nervous system tells skeletal muscle fibers what to do. So that's kind of a nice sum up of nervous system control in one cohesive GIF. Now we're going to pay attention to excitation. In order to understand excitation, we need to look at the neuromuscular junction. So the neuromuscular junction is the point at which a neuron communicates with a skeletal muscle fiber. This is a beautiful image of it. So you have your nerve, your somatic motor neuron right there that's part of a nerve. It branches out, right? And you see this interaction point between the muscle fiber and the nervous system. That point of interaction is called the neuromuscular junction. At the neuromuscular junction, action potential arrives at synaptic end bulb, which triggers the opening of voltage-gated calcium channels. Calcium influx mobilizes synaptic vesicles filled with acetylcholine. The acetylcholine is released onto the skeletal muscle fiber. Now, when you're looking at this structure right here, remember the plasma membrane of a skeletal muscle fiber is called the sarcolemma, and within the sarcolemma we have voltage-gated sodium and potassium channels, voltage-gated. But where the sarcolemma folds, right, you get this special region of the sarcolemma that doesn't have voltage-gated ion channels. It has ligand or chemically gated ion channels. The channels that you find there are nicotinic cholinergic receptors. So the channels that you find there are nicotinic cholinergic receptors. Where you find nicotinic cholinergic receptors, receptors for acetylcholine in the skeletal muscle fiber, right, is within a special part of the muscle fiber called the motor end plate. So the synaptic end bulb, aka terminal bouton, aka axon terminal, plus the motor end plate of the skeletal muscle fiber make what's called the neuromuscular junction. Now we've talked about EPSPs, IPSPs, which are a form of graded potential. We've talked about receptor potentials, which are a form of graded potential. Graded potentials that take place in a skeletal muscle fiber, meaning potentials that don't hit threshold, are called receptor potentials. So the reason receptor potentials occur is you're not always going to fire an action potential in the skeletal muscle fiber. Firing an action potential in the skeletal muscle fiber is called excitation. Skeletal muscle fibers, cardiac muscle fibers, smooth muscle, they all need to fire an action potential because that's what cues the mechanism of contraction. So excitation, which is the action potential in the skeletal muscle fiber, is what cues contraction, is what allows contraction to occur. And we call it excitation-contraction coupling. So let's look at what happens with excitation because it should sound pretty darn familiar. Action potential arrives at synaptic end bulb of somatic motor neuron. That opens up voltage-gated calcium channels. Calcium rushes into the cell, triggering the exocytosis of acetylcholine. Acetylcholine binds to acetylcholine receptors, which are nicotinic cholinergic receptors in the motor end plate of the skeletal muscle fiber. Nicotinic cholinergic receptors are really just ligand-gated sodium channels. They bind, sodium rushes in. If enough sodium rushes in to bring the cell to threshold, voltage-gated channels, and specifically sodium channels in the sarcolemma, activate, and they fall like dominoes. When voltage-gated channels activate, you get the firing of an action potential. God, it's almost like this comes up over and over again. Right? You get the firing of an action potential, which is going to sweep along the sarcolemma. So that's excitation. Now, you've seen this particular diagram again and again. What we're looking at now is not the communication with one neuron and another neuron. What we're looking at is the communication of a somatic motor neuron with a skeletal muscle fiber. Here we have the sarcolemma, which is a specialized name for the plasma membrane of a skeletal muscle fiber, and the folded region that actually interacts with the neuron and has receptors for acetylcholine or nicotinic cholinergic receptors is called the motor end plate. Action potential arrives at synaptic end bulb of somatic motor neuron, which activates voltage-gated calcium channels, triggering the exocytosis of the neurotransmitter that stimulates skeletal muscle contraction, which is called acetylcholine. Acetylcholine binds to nicotinic cholinergic receptors, which are really ligand or chemically gated sodium channels. Sodium channels open, sodium rushes into the cell. If the cell's brought to threshold, voltage-gated channels along the sarcolemma will activate. Now, there are a few small distinctions between an action potential that takes place in a skeletal muscle fiber and an action potential that takes place in a neuron. First off, the skeletal muscle fiber, due to the number of potassium leak channels and the negatively charged ions in the cell, has a slightly more negative resting membrane potential than a neuron. So if you look at the RMP here, it's negative 85. If you see negative 85 or negative 80, that's enough to tell you that you're not talking about a neuron, you're talking about muscle. And for this class, the only muscle physiology that we've gone over is skeletal muscle physiology. So you know you're looking at a skeletal muscle fiber. And then the process of the action potential is much the same. You have a receptor potential. If it gets to threshold, it activates voltage-gated channels located along the sarcolemma, right? So the opening of voltage-gated sodium channels allows sodium to rush into the cell, and you get the depolarizing phase of the action potential. At about positive 30, right, voltage-gated sodium channels deactivate or close and voltage-gated potassium channels open. You get the repolarizing stage. You don't get the after hyperpolarization because the cell is already quite hyperpolarized. Sodium-potassium pump kicks in, reestablishes that ionic disequilibrium. You're good to go. The action potential that travels along the sarcolemma of a skeletal muscle fiber is called excitation. So if you look at this particular diagram, here we have our somatic motor neuron, the axon terminal, aka the synaptic end bulb, and here we're looking at the muscle fiber. So you're seeing the muscle fiber here, right? Here we're looking at what's called a T-tubule, those invagnations of the skeletal muscle fiber that allow it to interact with the sarcoplasmic reticulum, specifically the terminal cisternae of the sarcoplasmic reticulum. So let's trace excitation to the point that it transitions into now looking at contraction. Action potential travels along somatic motor neuron until it reaches the synaptic end bulb, aka axon terminal, aka terminal knob, aka terminal bouton. I'm going to use synaptic end bulb. That opens up voltage-gated calcium channels. They're simplifying this image. Calcium rushes in, and that triggers the exocytosis of the neurotransmitter acetylcholine, which you can denote with capital A, capital C, lowercase h. Acetylcholine crosses the little cleft and binds to nicotinic cholinergic receptors in the motor end plate of the skeletal muscle fiber. The motor end plate is a specialized structure that serves a very similar functional role to a dendrite. It receives information in the form of neurotransmitter. The binding of acetylcholine to its receptor opens up the channels. Sodium rushes in. If enough sodium rushes in, it's going to trigger an action potential. That action potential is going to travel along the sarcolemma, spread down the T-tubules. The change in voltage along the T-tubule is going to activate what's called the DHP L type, L as in Lucy type voltage-gated calcium channel, which is mechanically linked to a ryanodine receptor, right, which is a calcium channel within the sarcoplasmic reticulum. When the voltage changes and activates this voltage-gated calcium channel, which is an L-type voltage-gated calcium channel, it changes its shape and there's a protein connection that pulls open the ryanodine receptor and calcium's released. Calcium is necessary for muscle contraction. So the link between excitation, the action potential in a muscle fiber, and the actual contraction that takes place is you need the action potential to trigger the release of calcium, and calcium is what's necessary for muscle contraction to occur when you're in the muscle fiber. So now that we've gone over excitation, which is essentially the action potential that travels along the sarcolemma, the plasma membrane of the skeletal muscle fiber, projects down into the T-tubules, DHP channels essentially open up ryanodine receptors, and you get a flux of calcium into the skeletal muscle fiber, and that calcium triggers the mechanisms of contraction. Now we're going to focus on contraction. You can't talk about muscle contraction without talking about the sarcomere, because the sarcomere is what facilitates muscle contraction. So remember, myofibrils are broken up into many units of these little structures called sarcomeres, and every skeletal muscle is made up of tons and tons of sarcomeres. So from Z disc to Z disc is a sarcomere. Skeletal muscle contracts using a mechanism called the sliding filament mechanism. It's called the sliding filament theory of skeletal muscle contraction. So what happens is the myosin heads bind to actin, and what they do is they pull, those myosin heads pull the actin or the thin filament closer toward the M line. When the myosin heads pull the thin filament closer toward the M line, the Z discs move closer together. The I band, which is the region that's just made up of thin filaments, gets smaller. So the I band shrinks substantially because all those thin filaments have been pulled toward the M line. And in a contracted sarcomere, the H zone completely disappears because now there's no region that's just thick filaments. When thick filaments pull thin filaments closer toward the M line, what happens is the Z discs of the sarcomere move closer together. So the sarcomere gets shorter. The sarcomere, because those filaments also overlap one another, also get thicker. So when you contract a muscle, it gets shorter and it gets thicker. I know that's difficult to see that thickness there, right? But it gets shorter and it gets thicker. That's the consequence of millions of sarcomeres simultaneously getting shorter and thicker. When millions of sarcomeres get shorter and thicker, the entire muscle gets shorter and thicker. So what we're looking at is what's called the sliding filament mechanism of skeletal muscle contraction. This is just a different way to look at it. This is there's a bit more movement here. So you have your Z-disc, over here. From Z-disc to Z-disc is a sarcomere. When skeletal muscle contraction is triggered, myosin heads bind to actin. They pull thin filaments closer toward the M line. These Z-discs move closer together. The sarcomere gets shorter. When millions and millions of sarcomeres simultaneously get shorter, what you have is a muscle contraction. The muscle gets shorter and thicker. So this is just a nice way to look at this sliding filament. The filaments themselves don't get shorter, right? It's just they slide over one another because that's how they're arranged. And that's what produces that banding pattern, if you remember from the previous lecture with the light band and the dark band. It's just I band, A band. So if we're going to look at the mechanism of contraction a little bit more carefully, let's look at it here and let's arbitrarily start at step 1. So let's de-abstract this just a little bit. Here you have your thick filament, right? And here you have your thin filament. Your Z-disc would be over here and your M line would be over here. And we're going to look at what's necessary for contraction to occur. So in order for contraction to occur, myosin heads need to bind and hydrolyze or break down ATP. The energy from hydrolyzing or breaking down ATP is used to cock the myosin head, almost like the hammer on a gun. The myosin head then has a bunch of potential energy. It's like a rubber band that's been pulled back. It's just waiting to go. Assuming that calcium, which are these two little purple balls right here, is elevated around the myofibril, that calcium binds to troponin, shifts tropomyosin, and exposes myosin binding sites on actin. When myosin binding sites are exposed, the myosin heads bind to actin, and the thick filament binds to the thin filament. So myosin heads bind to actin. When myosin heads bind to actin, we call that the formation of a cross bridge. Once a cross bridge is formed, you get the power stroke, where the myosin head pulls the actin closer toward the M line. The more myosin heads there are bound to actin, the greater the force of the contraction is going to be, right? So those are the 3 steps of contraction. After contraction, you need to have relaxation. In order for relaxation to happen, you need ATP. ATP, which is energy, right, binds to the myosin head, and myosin detaches from actin. So in order to relax a muscle, you need ATP. In order to contract a muscle, you need ATP. Myosin is one area where you need ATP. We're going to look at the 3 areas where you need ATP at a later point. But myosin is one area. The sodium-potassium pump is another, and calcium pumps are another, if you just want to know it. Right? So there are 3 discrete areas where ATP is being used in large quantities. So you need ATP to detach the myosin from actin. That's what ATP does initially. It binds in its complete form, and the binding of ATP to the myosin head allows myosin to detach from actin. And then you hydrolyze that ATP, cock the myosin head, calcium binds to troponin, shifts tropomyosin, the myosin head binds to actin, power stroke occurs, and then it occurs all over again. I want you to think about this question as you're going through this, because this is always a question I give on the exam, and I usually give it as a short answer question, and you may be in a semester where we're doing short answer questions. But if I asked you the question, rigor mortis is a condition that after death, people essentially, their muscles lock up. So their muscles remain in a consistently contracted state. And we call that rigor mortis. Now, knowing what you do about ATP and its role in not only muscle contraction, but muscle relaxation, why do you think rigor mortis happens? Why does rigor mortis happen, thinking about ATP being necessary to detach myosin from actin? Now we're going to watch this video and I'm going to walk you through this. So here's just a slightly different orientation. Here's our thick filament, and over here would be our M line. Here's our thin filament, over here would be our Z disc, and what you are looking at right here is the myosin head. And what's just happened is a muscle contraction has occurred, so we're going to start off by detaching that myosin head from actin.
With calcium elevated around the myofibrils, myosin binding sites on actin are exposed, allowing interaction of the myosin cross bridges with actin. Release of the myosin cross bridge from actin requires binding of ATP. After ATP binds,
ATP binds, myosin detaches from actin, muscle relaxes. ATP hydrolyzed, cock the myosin head.
Split to ADP and inorganic phosphate. The energy derived from this reaction is utilized to cock the cross bridge in preparation for the power stroke. The power stroke is initiated.
Boom! The moment that myosin binds to actin, that's called the formation of a cross bridge, and then when myosin pulls actin, that's called the power stroke.
When the myosin cross bridge binds to actin, the cycle begins anew when another ATP binds to myosin. At rest, most cross bridges are in the cocked configuration, prepared to interact with actin when the tropomyosin-troponin complex is moved away from myosin binding sites on actin by calcium ions.
Those are the biggest calcium ions in the history of calcium, and if calcium was really that big comparative to troponin, it'd probably fill up the solar system. But they're emphasizing a point. So calcium is released from the sarcoplasmic reticulum once an action potential travels down the T-tubule. So action potential down the T-tubule triggers the release of calcium from the sarcoplasmic reticulum. Excitation done. The link between excitation and contraction is the release of calcium. So excitation, the action potential in the skeletal muscle fiber, is what triggers the release of calcium. That calcium then comes and binds to troponin, and it shifts. When calcium binds to troponin, it shifts tropomyosin out of the way so myosin can bind to actin. In other words, muscle contraction can't happen unless there's calcium. And the only way that calcium can be released in the cell is if the cell fires an action potential. Skeletal muscle fiber fires an action potential. That triggers, when the action potential travels down the T-tubule, the release of calcium. Calcium binds to troponin, shifts tropomyosin out of the way, allowing myosin to bind to actin. The more calcium there is, the more troponin is bound, the more tropomyosin is shifted out of the way, the more myosin can bind to actin. Each individual myosin that binds to actin, each contact point, increases the force of a muscle contraction. So if you want to increase the force of a muscle contraction, you figure out how to release calcium, because more calcium equals stronger muscle contraction. Okay, so let's pull it all together. You're reaching for that water. So you have urine, sulfuric acid, and water in front of you. You look at that, you hear the instruction, take a sip. You go, oh, probably not urine, probably not sulfuric acid, but I'm going to go with that water. So information is bouncing around your brain, makes its way to the premotor cortex. Premotor cortex communicates with the primary motor cortex because it plans skeletal muscle contractions out, and it lets the primary motor cortex, hey, this is what we need to contract. Primary motor cortex communicates with the basal nuclei. That's a simplified version of what's happening, as does the substantia nigra, which can modify things in there. The basal nuclei communicate with the thalamus. Thalamus communicates back with the primary motor cortex. Upper motor neuron, which forms part of the corticospinal tract, communicates with somatic motor neuron. Somatic motor neuron communicates with skeletal muscle fiber at the neuromuscular junction. At the neuromuscular junction, action potential arrives at the synaptic end bulb of a somatic motor neuron, which opens up voltage-gated calcium channels. Calcium floods into the cell, triggering the exocytosis of the neurotransmitter acetylcholine. Acetylcholine binds to nicotinic cholinergic receptors located in the motor end plate of the skeletal muscle fiber. The binding of acetylcholine to nicotinic cholinergic receptors, which are really just ligand or chemically gated sodium channels, opens up sodium channels. Sodium rushes in. Sodium has a higher concentration outside the cell than inside the cell because of the sodium potassium pump. That triggers a receptor potential. If the receptor potential hits threshold, got an action potential. The action potential traveling along the sarcolemma of the skeletal muscle fiber is called excitation. If the action potential spreads down little folds in the muscle fiber called T-tubules, it activates what are called L-type voltage-gated calcium channels. They're also called DHP channels. Those are linked mechanically to ryanodine receptors in the actual sarcoplasmic reticulum, so it tugs them open. Calcium is released from the sarcoplasmic reticulum. Calcium binds to troponin, which shifts tropomyosin, allowing myosin to bind to actin, forming a cross bridge. After the cross bridge forms, the power stroke occurs, so the myosin pulls the actin closer toward the M line. The Z discs get closer together. The I band gets smaller. The H zone disappears, meaning that the sarcomere gets shorter. Millions and millions of sarcomeres getting shorter all at once mean the skeletal muscle gets shorter, right? So ultimately, skeletal muscles are just bundles of sarcomeres and other ancillary components. Now, after a muscle contraction occurs, you need to relax the muscle. So you got to clear out all of that calcium using these things called Circa pumps or calcium ATPases. So you have these pumps in the sarcoplasmic reticulum that use ATP directly to pump calcium back into the sarcoplasmic reticulum against its concentration gradient. So not only do you need ATP to cock the myosin head, you need ATP to fuel these calcium pumps, and of course you need ATP for the sodium potassium pump. Those are the three areas where ATP is used predominantly by a skeletal muscle fiber. Once the calcium is sucked up, the troponin tropomyosin complex shifts back into place, the muscle relaxes, and all of this accessory acetylcholine in the synaptic cleft, right, that could be binding to the motor end plate is broken down by acetylcholinesterase, which is the enzyme that breaks down acetylcholine. When you think about the exam, think about a question like this. This sums up excitation contraction coupling, right? Ultimately, the nervous system controls the muscular system. Action potential along the skeletal muscle fiber triggers the release of calcium, which ultimately leads to the contraction of the skeletal muscle fiber. That is excitation contraction coupling in a nutshell. So we're going to watch this video, and then we're not going to watch that video, and we're not going to worry about it. So I'll go ahead and show you this little Myasthenia Gravis clinical connection. I'm sure you guys are happy that the video didn't play, but I am not going back.